Up to this point, we've been discussing atoms and molecules. Now we will move up another level of organization to discuss organelles and cells. Microscopes were first used in the 1600s. Before that, we couldn't see cells, so we didn't understand the biological processes that take place in cells. The microscope opened up a whole new field of cellular biology. The first type of microscope was the light microscope. This is what you use in lab. This typically consists of a tube with one or two lenses that the observer looks through. There is a light at the bottom that shines through an object. We usually need the object to be very thin so that the light can come through. This process of light shining through a specimen is called transmission. It gives an image on the inside of something, like the picture of the paramecium on the far left. The lenses bend the light and magnify the image of the specimen. Light microscopes can only really magnify something oh so much. Eventually, the resolution becomes very poor. The resolution is how clear an image is. If you've ever tried to zoom in on a picture and found that you can't see the image anymore, this is due to poor resolution. This limits the size of specimens that we can see on a light microscope. Typically, we can see things like small cells, as bacteria are very small, and maybe even some of larger organelles. Another type of microscope is the electron microscope. This sends a beam of electrons at a specimen, and they bounce off or go through it and create an image. With scanning electron microscopes, the beam of electrons bounces off the surface and creates a 3D image of the surface of the specimen. You can see a scanning electron microscope image of a paramecium in the center picture. A transmission electron microscope uses the transmission of electrons through a specimen. This means that they shoot a beam of electrons at an object, but instead of bouncing off, they go through, creating an image of the inside. The picture on the far right is a transmission electron micrograph of a paramecium. It has a lot more detail of the inside of the paramecium, and the resolution is a lot better than the light microscope image. Electron microscopes have several drawbacks, though. They are very expensive, and they cannot be used on living things. All the methods used to prepare specimens for electron microscopy will kill them. The use of microscopes has allowed scientists to develop what we call the cell theory. The cell theory says that all living things are made up of cells, and that all cells come from pre-existing cells. You've probably noticed that all of the cells we talk about are very small. There are no gigantic cells like the blob running around on Earth. Cells would not be able to function if they were very large. Cells have to be able to transport materials that they need across the plasma membrane all the way through the cell. Cells are restricted in size because as they get bigger, the plasma membrane cannot keep up with the needs of the cell. This is due to something called the surface area to volume ratio. A ratio is just talking about division. So the surface area to volume ratio is the surface area of the cell divided by its volume. The surface area of the cell is represented by its plasma membrane, and the volume is all of the liquid cytoplasm on the inside. The plasma membrane is a bilayer of phospholipids that surrounds the entire cell. We usually think of cells as spheres, but they can be many different shapes. For this example, let's pretend our cell is a cube. A cube that has dimensions of 1 by 1 by 1 would have a surface area of 6 and a volume of 1. This means that 6 divided by 1, the surface area to volume ratio, would be 6. Cells need to have a large surface area to volume ratio. Look in the screen at the example of the large cell. It is a 3 by 3 by 3 cube, which has a surface area of 54 and a volume of 27. This results in a surface area to volume ratio of only 2. This cell is much larger and will have a harder time getting things like oxygen to all of its organelles. You can see the link at the bottom to watch a YouTube video about an experiment using different sized cells, which shows how larger cells are not as efficient at getting nutrients. There are two main types of cells, prokaryotic and eukaryotic. Prokaryotic cells are things like bacteria, such as kingdom Eubacteria and Archaeobacteria. All other cells are eukaryotic, such as animals, plants, fungus, and protists. Let's take a look at these words. The prefix pro means before, and karyo means nucleus. So prokaryotic means before nucleus. The prefix eu, EU, means true. So eukaryote means true nucleus. This shows one of the main differences between these types of cells. Prokaryotes don't have a nucleus. The DNA is circular and is clumped up in a nucleoid region in the center of the cell, but it's not surrounded by a membrane. Eukaryotic cells have a true nucleus with a nuclear envelope membrane and many chromosomes inside. Prokaryotic cells do not have any organelles with a membrane at all. These are called membrane-bound organelles. Eukaryotic cells have a lot of membrane-bound organelles. Prokaryotic cells are believed to have evolved first. It is likely that eukaryotic cells evolved from an ancestor that was a prokaryotic cell. Prokaryotic cells are also much smaller than eukaryotic cells. We're going to focus on plant and animal cells for the rest of the chapter. These are eukaryotic cells. Plant and animal cells have a few key differences that I want to highlight. There are specific organelles that appear only in plants or only in animals that help us show the kind of functional differences between these types of organisms. Plants have chloroplasts for photosynthesis, a large central vacuole for storing water, and a supportive cell wall. Animal cells don't have any of these three organelles. Animal cells do have centrioles for cell division and lysosomes for digestion that plant cells don't have. One good technique for studying these last two slides of similarities and differences is making Venn diagrams. Try to make one for the differences between prokaryotic and eukaryotic cells, and then make another one for plants versus animal cells.